Welcome to Module 1 of Enzymes and Metabolism. In this part of the course, we are going to explore one of the most fascinating areas of biochemistry, enzymes. Enzymes are the unsung heroes of life. Without them, the complex chemical reactions that keep cells alive would either happen too slowly to sustain life or not happen at all. Imagine a crowded marketplace, traders shouting, customers bargaining, goods being exchanged. This chaos represents the constant activity inside our cells. But in this busy marketplace of molecules, enzymes act like efficient store owners who know exactly what their product is, where to get it, and how to deliver it, quickly and precisely. Enzymes are biological catalysts. That means they speed up biochemical reactions without being consumed or permanently altered in the process. They make life possible by lowering the energy barrier, known as the activation energy, that would otherwise make reactions too slow to be useful. Classification of enzymes, the 6 major classes. The International Union of Biochemistry and Molecular Biology, IUBMB, classifies enzymes into 6 major classes, based on the type of chemical reaction they catalyze. Let's go through them, one by one, with simple examples to help you remember. 1. Oxidoreductases. These enzymes catalyze oxidation-reduction reactions, where electrons are transferred from one molecule, the donor, to another, the acceptor. Think of them as electron movers. Example, lactate dehydrogenase, which converts lactate to pyruvate by removing hydrogen atoms. 2. Transferases. These enzymes transfer a functional group, such as a metal or phosphate group, from one molecule to another. Picture them as parcel delivery agents, moving packages from one address to another. Example, alanine transaminase, which transfers an amino group from alanine to alpha-ketoglutarate. 3. Hydrolases. These enzymes break chemical bonds by adding water, a process called hydrolysis. They are like molecular scissors that cut big molecules into smaller ones. Example, amylase, which breaks down starch into simpler sugars. 4. Lyases. Lyases break bonds without using water or oxidation, often forming a double bond or a new ring structure. Think of them as molecular rearrangers. Example, pyruvate decarboxylase, which removes carbon dioxide from pyruvate to form acetaldehyde. 5. Isomerases. These enzymes rearrange atoms within a molecule to form isomers, molecules with the same formula but different structures. Imagine a Lego structure being taken apart and rebuilt in a new shape, using the same blocks. Example, phosphoglucose isomerase, which converts glucose 6-phosphate to fructose 6-phosphate. 6. Ligases. Ligases join two molecules together, usually with the help of ATP. They are like molecular glue. Example, DNA ligase, which joins DNA fragments during replication. Factors affecting the velocity of enzyme-catalyzed reactions. Just like human workers, enzymes have optimal conditions under which they perform best. Change these conditions too much, and you can slow them down, or even stop them entirely. Effect of substrate concentration. Imagine you are running a restaurant with a fixed number of chefs, enzymes. If you only have a few customers, substrates, the chefs work quickly and efficiently, finishing orders in no time. But as more customers arrive, the chefs get busier, and the time to serve each order increases. Eventually, every chef is working at full capacity. No matter how many more customers you add, the service rate cannot increase. This maximum speed is called Vmax, the maximum velocity of the reaction. Initially, increasing substrate concentration increases reaction velocity, but once all the enzyme active sites are occupied, the reaction reaches saturation. Effect of enzyme concentration. If you double the number of chefs in your restaurant while keeping the number of customers constant, service speed doubles, because more chefs can work at the same time. Similarly, if the amount of enzyme is increased while substrate is abundant, reaction velocity increases proportionally. However, if there's not enough substrate, adding more enzymes won't help. Effect of temperature. Enzymes are proteins, and proteins have delicate structures. At low temperatures, enzyme activity is slow because molecules move sluggishly and collide less often. As temperature rises, activity increases, up to an optimal point, because molecules collide more frequently and the enzyme's flexibility allows better binding to the substrate. However, Beyond the optimal temperature, heat begins to denature the enzyme, unraveling its structure, and the activity drops sharply. This is why a high fever can be dangerous. Many enzymes stop functioning efficiently when body temperature climbs too high. Effect of pH. pH affects the charges on amino acids that make up the enzyme, particularly those in the active site. If the pH is too acidic or too alkaline, it can disrupt the bonds holding the enzyme together or alter the shape of the active site, reducing efficiency. Each enzyme has its own optimal pH. For example, pepsin, an enzyme in the stomach, works best at a very acidic pH of around 2, while trypsin, in the small intestine, prefers a pH of around 8. Michaelis-Menten equation and Km. The Michaelis-Menten equation mathematically describes how enzyme activity depends on substrate concentration. While we won't write out the equation here, the key idea is that it relates reaction velocity to substrate concentration, maximum velocity, Vmax, and a constant called Km. Km, the Michaelis constant, is the substrate concentration at which the reaction reaches half of its maximum velocity. A low Km means the enzyme binds substrate very tightly, it doesn't need much substrate to get going. A high Km means the enzyme binds substrate less efficiently, it needs more substrate to work at half capacity. The Lineweaver-Burk plot. The Lineweaver-Burk plot is a graphical method to determine Vmax and kilometer. Instead of plotting velocity against substrate concentration, you plot the reciprocals, 1/velocity against 1/substrate. This transforms the curve of the Michaelis-Menten equation into a straight line, making it easier to extract values for Km and Vmax. Cofactors and coenzymes. Not all enzymes can function alone. Some need helpers. Cofactors are non-protein components required for enzyme activity. They can be metal ions like zinc, magnesium, or iron. Coenzymes are organic molecules, often derived from vitamins, that help enzymes. Examples include NAD from niacin and FAD from riboflavin. Think of the enzyme as a machine. The cofactor or coenzyme is a necessary part that makes it run smoothly. Enzyme specificity. Enzymes are incredibly selective. Their specificity can be categorized as follows. Group specificity. An enzyme acts on a group of related substrates. Example, hexokinase can phosphorylate several different hexoses, such as glucose and fructose. Optical specificity. An enzyme distinguishes between optical isomers, molecules that are mirror images. Example, L-amino acid oxidase acts only on L-amino acids, not diamino acids. Geometrical specificity. An enzyme recognizes differences in the arrangement of atoms in a molecule. Example, fumarase acts only on fumaric acid and not on its cis isomer, maleic acid. Cofactor specificity. An enzyme requires a very specific cofactor to function. Example, carbonic anhydrase specifically requires zinc as its cofactor. We've covered the basic but essential knowledge of enzymes, their classification, the factors that affect their activity, the concept of Km and Vmax, the Lineweaver-Burk plot, the importance of cofactors and coenzymes, and the various types of enzyme specificity. These concepts will serve as the foundation for Module 2, where we'll explore how enzymes drive the complex pathways of metabolism, the processes that extract energy and building materials from the food we eat.